Hi, my name's Colette Quinn. I'm the microcalorimetry product manager at TA Instruments, and today I'll be giving a seminar on antibody drug conjugates, a DSC case study. This case study is made possible through contributions of samples through my colleagues at Waters, Weibin Chen and Henry Schoen, and I just want to say thanks to both of those before I get started and before I share the data that was collected. So first of all, to get everybody on the same page, we have to talk a little bit about antibody structure and the regions that make up an antibody. So antibodies demonstrate a strong structure-function relationship. And because of this relationship, the unfolding or denaturation temperature measured by DSC is associated with functional differences. It's made up of three main domains. One is the FAB domain, that's the upper portion that make up the two upper regions of the Y. And then you have the CH2 domain and CH3 domain. So the FAB domain is the antigen binding region. So that's where it actually finds the antigen and would bind it. And then you have the crystallizable regions, the FC domain, that are the CH2 and the CH3. So it's good we all have an idea of the majority part of the molecular weight of an antibody comes from the FAB, and then the two minor pieces, the CH2, CH3, those are lower molecular weight domains. And this is going to play into the DSC thermogram and what we see. So now starting off on what is DSC, what kind of information can we get from a DSC thermogram? What we're measuring on DSC is the heat capacity. So the components that you can actually get out of a DSC thermogram are the change in heat capacity, where you've got a heat capacity before and after to get the delta Cp. Typically this is a positive value due to an increase in flexibility, hydrophobic exposure. The main part, though, is underneath the red unfolding trace that you can see in the picture, and that's how we get the enthalpy. And the enthalpy comes from the energy of the non-covalent interactions. These could, or even hydrogen bonding interactions. A lot of these hydrophobic and electrostatic interactions. So all the energy that makes up holding a protein together. And that also includes solvation effects as well. And then finally, you have the TM, the most commonly reported component that you get out of a DSC thermogram, which is an indicator, sometimes inferred to as the macromolecular stability. Folding and unfolding of a small protein or a simple domain or subunit, as we're going to talk about in the antibody, it's got those three domains, is cooperative, meaning once it starts, it goes to completion. So typical antibody thermograms, really the thing that makes an antibody thermogram typical is that it is atypical. So you're going to see different variation in the thermograms, whether it's an IgG, as the one shown here, or an IgA. So in the two IgG examples that we have here, we have the first unfolding event, which is attributed to the CH2 domain. You have the large event that has about a Tmax coming in about 67 C, and that's the FAB domain. And then finally the high temperature unfolding event, which is the CH3 domain. Now when these domains are overlapping, so meaning overlapping in the stabilities, as shown in the lower thermogram, you can still deconvolute and then assign. It's very typical in the unfolding of an antibody that you have CH2, FAB, CH3 in that order. So really what I want to introduce now is kind of a new model within our software, but also a very simple model that can be used to describe proteins. So there are a few models that are currently used to describe proteins, the most common one being the two-state model, meaning that you have either a native or a denatured form, or native or an unfolded form. Now, one of the assumptions with these kinds of models, these two-state scaled or these pseudo-Van Hoff models, is that they are, at least on the very least, microscopically reversible. The nice thing about the Gaussian model is that we're not making any assumptions to reversibility, which is anybody that's worked with an antibody, once you get past the CH3 domain, once you get past the CH2 domain, it's really unlikely that you're going to have reversible unfolding. So with the Gaussian model, you still get descriptors of the unfolding. You're going to get a max or a Tmax value. You'll get an amplitude value, so just the height, and you'll get the area value, which is the enthalpy. And then the final component or the final descriptor of it is this full width at half max value. So the full width at half max value is a good indicator actually of cooperativity. So if something's very narrow, we can consider it very cooperative in its unfolding. Kind of an easy way to think about this is in terms of organic chemistry. So you've got a molecule that you just made in the organic chemistry lab, you bring it over to the melt temp instrument, and you really hope that that melt occurs immediately, and then when you cool it back down, it becomes a crystal again immediately, meaning that you have one crystalline form. And this is true of any kind of material sciences, that you want a very sharp unfolding peak. Well, the same idea could be kind of applied here to the proteins. So if you've got a very broad unfolding, we've lost some cooperativity, but really the idea is that we've got a protein in multiple forms. Definitely we're always going to have a protein in multiple forms because it's very dynamic in solution. But in here you've got multiple energy or degeneracy levels. So you've got some that could be a lower energy and some higher energy, but then the majority fitting in the middle, just like the Gaussian curve describes, right? Like a bell curve or a distribution curve. So we'd really ideally want everything to be in a narrow curve, because then we at least know we've got majority in native or unfolded. And as we start to spread out, we have the possibility of that intermediate or that stable intermediate being available in solution as well, which then we have questions of, is that intermediate the right form to then find its right target? So I'd say the Gaussian model, nice for a few reasons. One is that simplicity in it, and then two is that extra full width half max value that will give you an idea of cooperativity unfolding and the possible presence of an intermediate form. So what are antibody drug conjugates? I went to an ADC conference a few years ago, and somebody put up this picture of this the lion sidecar wall of death, and I thought it absolutely described antibody drug conjugates really well to somebody maybe just very new to the field, or somebody that already knows it and appreciates kind of the struggles of antibody drug conjugates. So here's the idea. The car and the motorcycle, those are your antibodies. And then attached to your antibody, you have a highly cytotoxic drug. That's the lion. So the idea is that you want your car is very directed in its motion to chaperone the lion to the finish line or to the target, in our case of antibodies, right? So the antibody is going to be very specific of finding the target, and then it's going to deliver the lion. But it's really important that that lion doesn't get loose in the course of a race or in the course of moving through your body, and it doesn't go and attack all the civilians on the sideline, or it doesn't go and attack other things to cause other mal effects or other bad effects in your body. The other important thing of this picture, what it shows, is that you have a lion attached to one of the cars, but not to the other, not to the motorcycle. And this would be very ineffective in its delivery of this antibody drug conjugate. Really, you'd want a lion sidecar attached to every single antibody that you have prepared running through there, so you have the right dosage of that ADC. So ADCs, they've definitely, you can see the challenges here in the picture, but there has been an increase in ADCs in the market. So in 2016, there are 2 licensed ADCs that were available. The first one being the brentuximab vedotin, and this was a drug released by Takeda and Seattle Genetics. And then the other one is the trastuzumab with the DM1. So the trastuzumab, that's a Herceptin antibody with that cytotoxic drug attached to the side chain, and that's by Genentech and Roche. So we've got 2 in 2016 licensed ADCs on the market. And if we really look at what is the global market for this, well, the global market for an antibody drug conjugate was valued at $1.3 billion in 2016, with an increase of a 20% growth until 2021. So definitely a growing market and a high demand for these types of specific drugs and highly toxic drugs as well. So we think about how are they doing before they enter the market. In 2015, there were 17 novel ADCs entering clinical testing for the first time, with a similar trend identified in 2016. So definitely there is a demand for these drugs, and they are being developed by the pharmaceutical companies. So really, just thinking about, again, okay, now let's actually look at a cartoon drawing instead of the lion in the sidecar. What does an ADC look like? So in this cartoon, we have, on one side, we've got just the the antibody with the DM1, that cytotoxic drug attached to it with a thioether linker. And then the other side, we can actually see that specific cytotoxic drug. So Herceptin is approved for use only in human epidermal growth factor receptor, or HER2 positive cancers. But not all HER2 positive cancer cells have sufficient apoptotic capacity to be killed by trastuzumab via binding alone. So this is where the ADCs really come into play here, is that you've got this highly specific antibody. It's going to go find that target, find the HER2, and then it's going to deliver its toxic load. So whereas it would find the target and it wouldn't lead to apoptosis, now that we've got this toxic molecule on there, it can actually go in and deliver it, and that cytotoxin will actually bind to the tubulin to prevent microtubular formation. So really a very effective way of causing apoptosis. So one of the problems with ADCs is how many drugs do you have actually attached on there? There are a few different ways to attach these cytotoxic drugs onto their antibody. And the most common way, or the most commonly adopted way, and kind of the only adopted way now, tends to be the cysteine linker. So in an antibody, you have potential for multiple, the disulfides are broken down, and the antibody then is attached to that disulfide. So you have a possibility of 4 in the CH2 domain, and then you have 1, 2, 3, 4 up in the FAB domain. So you've got those 2 domains that could possibly be linked with this cytotoxic drug. Now the question is, though, is how many do we have linked on to there? Can we tell a difference between a low versus a high linkage of that drug? So can we tell that we have 2 linked on, 4 linked on, 6 linked on? Or at least have an idea of a low load versus a high load. So then we have an idea of efficiency of that drug being attached to the antibody. So this is an example of a conjugation type 1. Generic, we'll just call it conjugation type 1 of an antibody drug conjugate in series. And it's run at 0.5 mg per mil. We can see at this low concentration, we could easily go as low as 0.1 mg per mil and still see very nice resolution in the thermogram. So here, let's just first look at the native profile. So the native is the profile in black, and we can see the very large FAB domain. We can see a slight shoulder, probably with a T onset coming in about 60-61 degrees for that before the first the CH2 domain, and then we can definitely see the CH3 domain following the large FAB unfolding. But when we start to add the drug, even at low load, moderate load, and high load, we can actually see a separate species start to form in solution, a low temperature species. So this low temperature species, it wasn't at all seen in the native form, and we can actually see it growing as we start to add more and more drug to it. So as we go from low to moderate to high. So definitely we could tell a difference there by the amplitude, if we're just thinking about a Gaussian model, right? The amplitude starting to grow over time, the enthalpy starting to grow over time. With the enthalpy starting to grow over time, then that gives us an idea, now again, just of the CH2 domain. With the enthalpy starting to grow over time, we have an idea then that possibly we have an increase in this separate species, this species that has a lower energy level than the typical CH2. Now, it is a mixed species. We still have a little bit of that shoulder showing up, overlaying with the FAB domain. But we definitely have a growth in this lower temp, lower stability species of the CH2 domain. So that's one way we could look at it. The other way, though, is let's just look overall at profile differences in the FAB domain. And the easiest way would be either, again, amplitude, or a more direct way would be percent enthalpy compared to the native. So why would this be important? All of these were thought to, via UV-Vis, to have the exact same concentration. But the question is, even though via UV-Vis we may have seen something, you may have seen a protein, was it in its folded form? So this is a good QC/QA check right here as well. So if we look at the percent enthalpy compared to the native in that last column, we can see as we add more and more drug, the enthalpy starts to decrease. So it's either one of 2 options. Either we're having unfolding of the protein, and we just flat out don't have as much antibody available, which is going to be a problem later on for, if we're talking about dosages, or we've actually just broken apart a lot of the bonds that actually held this antibody together. So it's one of two things that could be occurring here, but with this large percent enthalpy change, it's more likely that we had some denaturation of the antibody during the preparation step. So other things to look at, what other changes could be going on looking at this? What else can we get from the DSC thermogram? And one would be this full width at half max value that we talked about earlier. So if we're looking at just mainly the FAB domain, we can see a difference in the full width half max. It's actually becoming narrower in the FAB domain, meaning that we could potentially have an increase in cooperativity of that FAB domain. So they're all sitting now more into a decreased number of degeneracy levels, decreased number of energy levels. So we can see that through the full width at half max, or alternatively, another good indicator that actually starts to parse things out, not just from native to high, but incrementally through the native, low, moderate, and high, is the difference between the T max of the FAB and the T onset of the FAB domain. And we can start to see differences that would absolutely start to help us understand how to differentiate between a native load or native with no drug, low load, moderate load, and high load. So in addition to this, we could actually go ahead and fit all of the data and start to look at trends between each domain. But I think just the overall looking at it through that simple Gaussian gave us a good idea. But here, separating out each thermogram and fitting them, this is how we can really see that additional CH2 is what we're proposing that it is. That additional CH2 species start to grow, and then the decrease in all of the heats here. But really a similarity between the CH3 domain, FAB changing only slightly in the enthalpy, and really not changing at all on the Tmax value. So this kind of gives value to the DSC thermogram, or the shapes that you can get out of the thermogram. We're not just looking at a simple Tm value, looking at the difference between T onset, Tmax. We really start to see differences. So the deconvolution of the data and subsequent fitting with the pseudo-Van Hoff model allows for assignment of percentages of heat. You can also get these percentages of heat via the Gaussian model as well. But via the Van Hoff model, we actually simplified the analysis and just looked at total heat, which we'll call Q. So we take the delta H Van Hoff times this scaling factor that we have, that we added in order to fit the data. And normalizing the data allows a comparison across the entire series. So we start to think about that first peak that we saw, we'll call that Q1, and then we have Q2 plus Q3, that'd be the CH2 plus the FAB, and then Q4 being the CH3 domain. And we can start to look at percentages and get an idea that we do have growth that is occurring as we add more and more of the drug, and that growth is occurring to that separate low temperature species. But definitely the majority of the heat originates from the middle temperature transition, not surprising. That's where the majority of the molecular weight resides as well, with the FAB in the CH2 domain. And then we see that the low temperature event increases from non-existent in the native form to 9% at high. At 9%, 10%, that is definitely a significant change and a significant species shift that we can think about. And also the increase in the Q1 is linear. So it looks like how they're going on is very in a similar fashion, in that it's the same species, and we just have more and more of that same species growing as we add more and more of the drug. Okay, so now we can break it down. Just this is a different picture of just looking at that high load and then breaking it down into the Gaussian. And we can see a few differences between the two CH2s, where we've got one with a very narrow full width half max and the one with a wider full width half max. But just another way of looking at the data of deconvoluting via this other model and getting similar information that we did via the other type of analysis. So a few final comments on the conjugation type 1 data. So we had a Tmax decrease from native to high, a slight decrease. More than 0.3 degrees C is significant for how we calibrated this calorimeter. The width of the peak, the full width half max, decreased from native to high, and there was an increase also observed at the lower T onset and a slightly larger T max. The modified forms have a low temperature peak that is absent in the native form, and the peak's magnitude increases from low to high. And then we have an enthalpy decrease from native to high. The net enthalpy, as described before, comes from the direct breaking of hydrogen bonds or electrostatic interactions. either these bonds are not formed in the modified forms, or a small percentage of them, only a smaller percentage is actually correctly folded after we've added that drug load. Okay, so there's one type of conjugation. We saw some interesting trends in that. And now here's the second conjugation type. So this is a different antibody, so we can already see by the native profile, which is actually this black, the first black top one that has the very large peak 1 and peak 2 is what we'll just call it generically. Definitely a different unfolding, but not surprising because we first saw how some antibodies unfold differently. But let's see how this series looks as we go from a low to high load. So first we have the native, and we have a very large first peak, and then we've got the second peak with likely, with that little shoulder on the end that is likely the CH3 domain. But we're just going to look at differences between those first larger peaks. So as we add more and more drug, actually that first peak disappears. So a totally opposite trend of what we saw in the other conjugation type, where we have the absolute disappearance of that first initial peak, and now everything is just sitting under basically one unfolding transition. So not distinctive domains, no distinctive domains are really seen as we add more and more to the drug load. And unfortunately, that's all I can say about the conjugation 2 type without giving more away. But definitely some interesting trends without giving any more an idea, and maybe you'll have an idea right now in your head of what kind of conjugation type this is, as we can see it very broad in the thermogram. So let's look at a few other differences. One is the peak area. We can see that peak 1 absolutely going from 21% of the total area down to none of the area at a high load. And we really see no difference in the full width half max of that peak 2. And so that's the FAB domain. If we talk about enthalpies, there's definitely a decrease in enthalpy as we go from native to high. So a similar trend as we saw as we added more and more of the drug, possibly because we've seen it on 2 different types now, possibly backing up the idea that we do have some denatured protein in there as we start to add more and more of this drug. But really, if we want to talk about what kind of differences we can see and actually quantify, it's through the T max minus the T onset is where we actually see a difference, and also just the loss of the peak 1. So 2 different trends, but we can still fit it. We can still deconvolute just like we did with the other, and we can still think about assigning what each domain is in the thermogram. So the first being the CH2, and then the FAB, and then the CH3. And then eventually we have all of those different domains really coming in together, and really no differentiation between the domains, all just sitting under one unfolding thermogram. And just for simplicity, although we do know that there is a domain sitting under that large peak in the moderate and the high, I think it's best to be simple in analysis and just assign it 2 peaks, and then in that peak assignment talk about what domains actually lie under there. So comments on the fitted conjugation 2 type data. The majority of the heat originates from that second peak, the Q2, with an increase of the percent Q2, so the percent enthalpy, as the extent of modification increases. So really enthalpy giving us a nice insight into the differences between native low and then moderate high being very similar and hard to distinguish between the 2. This increase was concurrent with an increase in the width, which could indicate the extent of the conjugation of the type 2 modification actually stabilized this population. So they all started to come in very narrow, and they're all in a similar, well, with similar energies then and similar stabilization. For certain antibodies, the initial peak can indicate flexibility of the hinge region. It's possible that this flexibility decreases as the modification increases. It's kind of going off the idea of something like an IgA, where you have a very inflexible hinge region, and usually see an overlap between the CH2, FAB, CH3 domains. Possibly we've lost flexibility here as we've added this drug in this other conjugation type. So a few more comments on the conjugation type 2 data. We can see the Tmax decreased from native to high, but it was insignificant, so it was less than 0.3. So we really can't put anything on the small 0.1 degree changes. The majority of the species had similar stability. The width of the peak increased from native, or the width of the peak actually decreased from native to high, and this could indicate a greater level of homogeneity in the native sample, or it could come from stability of the low temperature species, increasing and overlapping with the stability of the middle species. The first integratable peak of the native form is absent in the later modified forms, and all of these start to fall under the one unfolding event. And then the enthalpy decreased from native to high, with the idea possibly of denaturation occurring as the drug was added to the antibody. So the one question is, what do you go from here? What do you do next, or what did you do first? And one of the ideas, because of my association, or because of TA's association with Waters, would be what did Waters do in this typical? What would Waters do in this kind of assay, or how would they assay antibody drug conjugates? And actually have a very nice recent paper talking about next step. So looking at a multiple platform analysis. And they did an LC-UV hydrophobic interaction chemistry, or HIC chemistry, with LC-LC QTOF-MS. So what they're able to do is they're able to say, okay, here under these UV unfolding, this UV spectral photometric assay, we can actually identify what those peaks are. So they took the peaks, they basically took what they call the heart section. So they took some of that solution out, and then they ran LC-LC QTOF-MS on it. And so then they're able to identify where in that UV profile what the positional isomers were. And if we look at the options for positional isomers, we can see a lot of them coming in at that CH2 domain for this particular conjugation, which would be a cysteine conjugation. So if we think back to both of those conformational, or both of those conjugates that we had on the DSC, definitely the CH2 domain is where we saw the large degree of change, the large degree of stability change in those. So very nice complementarity between this particular assay and then the DSC assay. The DSC gives you a nice place to look at the CH2 domain. That's definitely where some changes are at, and that could be followed up, or in addition to this very nice separation chemistry assay. And then I guess the other question is, where else do we go from here? What kind of other multiple platform analysis could you do? And absolutely the next step would be something like ITC, a native binding assay where you're looking at this particular drug, the Herceptin or the trastuzumab, with this DM1 attached to it, and then binding to the HER2 receptor. Do we see a difference in binding with a low or a high? Do you see a difference in the affinity? Do you see a difference in the enthalpy? So do things change in the binding assay as well? So that would be, one other place of where to go, just thinking about multiple platform analysis. So an ADC recap on the seminar given today. Type 1 and type 2 have different trends with increasing saturation levels. And then deconvolution and simple heat, or enthalpy assignment, really help describe the systems. And really this is a nice argument for DSC and QA and QC because of the differences it could see between these minor changes of a low, medium, and high load versus the native forms. Okay, thank you. So that is the end of the seminar, and now I'd be happy to entertain any questions that you might have.
All right, thanks. So we had a few questions that came in, and I'm going to go ahead and start with this first one. This is a question on how the drug load was quantified. So in the webinar, I presented a low, medium, high load. This was actually done not by me, but by collaborators at Waters Corporation. So if you're looking for the reference on that, it's in MABS Volume 7 from 2015. And that's Birdsall et al. And they're the ones that actually quantified via mass spectrometry what the actual drug loads were. All right, so here's another question. So the lower the T onset, the lower the stability, right? Yes, that's true. Or it's the general rule that people do. So the lower the T onset, the lower the stability, and that just has to do with, in a two-state situation, the T onset, but more likely the T max, is related to the concentration of the products unfolded over the concentration of the reactants. And so that's why they say lower the temperature, lower the stability. And then T onset will actually give you when all of this unfolding process starts. So you can actually get percentages of unfolded. So at the T max, if it is symmetrical, at that point you have half of your molecules unfolded. well, from T onset to T max, if you're just looking at percentages or the slices underneath of that unfolding thermogram, you can start to get an idea of the percent that's actually unfolded at certain temperatures. And then the other idea is looking at the difference from the T onset to the T max. And the larger the difference, the greater distribution of species or of things that are of species that have different stabilities in there. And then there's a follow-up question, actually on the same one, and is, did you look at any viscosity or structural changes? No, we did not in this study, so we didn't use a viscometer, which definitely gives some other interesting information, or a CD, but I would absolutely recommend both of these for greater understanding of this system. Okay, this next one is, was a TA-DSC used to generate the data? Yes, we used a capillary cell nano-DSC from TA Instruments. So this is our fixed cell nanocalorimeter that has a sample volume that we loaded in 550 microliter in the sample and the same volume into the reference. All right, so this next one is, do you have any stress stability data, for example, size exclusion chromatography, change after incubation at 40 degrees C, suggesting the lower the T onset, the lower the stability, or looking at the TM for the different domains, CH2, CH3, Fab, and which one is most related to stability? So we did not do size exclusion chromatography, but if you are interested in it, we do have another webinar that talks about stability measurements using isothermal calorimetry, and then linking those types of studies on our TAM or our isothermal calorimeter back to size exclusion chromatography data. And that was a 2018 webinar. As for the other part of the question, talking about which domain to look at for stability, there was a paper out recently by Brader et al., and where they looked at which domain is the most stable or what domain to look at. And they said because of reversibility, or typically there's the CH2 domains unfolding as reversible, that this one is not indicated for or not used in stability measurements when reporting. It's actually typically the FAB domain that's used in stability measurements, or is the more commonly accepted Tmax, T onset for stability of an antibody. All right, so I will make time for one more question. Okay, and this one is a user of the TA-Q20 instrument. So this is our traditional DSC from the thermal line, where they're looking at removable pans. And his question was, can I use the TA-Q20 DSC system to study proteins? Absolutely. So one of the things to consider, though, with that kind of instrument is the sensitivity of the instrument versus something like this nano DSC system. And it could be roughly about 100 times less than the nano DSC. So typically people are loading in 20 to 50 meg per mil to actually see the full unfolding. Now, the downside, though, is absolutely the geometry. Because we don't have the capillary where we're attenuating any kind of aggregation, it is possible that you are going to see aggregation immediately following the unfolding. Okay, well, that's all the questions that I have time for, and those additional questions that were submitted, I will make sure to follow up individually with everybody. I appreciate everyone for attending the webinar today.